OMG, let's revise 1.1 cells. Below are 7 core knowledge questions on this topic. Press pause on the video and attempt them. Here are the answers then. So question number 1, a root hair cell is adapted. It has thin walls, large surface area and no chloroplasts, not needed within the soil. 2, how do you calculate magnification? It equals eyepiece lens times objective lens. Diffusion, number 3, is the movement of particles from a high concentration to an area of lower concentration, down a concentration gradient. In 4, osmosis is the movement of water, again down a concentration gradient, but remember it's through a semi-permeable or selectively permeable membrane. Number 5, active transport, the movement of particles against a concentration gradient from lower concentration to an area of higher concentration, requiring energy. Number 6, the term enzyme is a protein and biological catalyst. It speeds up the rate of reaction without being used up itself. And finally, number 7, so an enzyme is affected by temperature. At higher temperatures, enzyme denature, the active site irreversibly changes shape. On to some past paper questions then. So pause the video, have a go at these ones on the screen, and then we'll go through the answers together. Okay, so here are the answers for this Viscon tube question. If we could have a look at the table, okay, at the point at which amylase was added after the hour, you can see that this is when glucose starts to be present. So that's what the first part was asking in part A. Explain the results. So 3 marks stating that amylase broke down the starch to the glucose for the second mark, and finally the third mark, which was then small enough to diffuse through the Viscon tube and into the water. Part B says state why starch was not found in the sample water, and it's to state that it's too big. Okay, so it's too big to pass through the Viscon tubing. In C, this model gut shown opposite, what does the water surrounding the Viscon tubing represent in the living body? And the answer is blood. And then finally at the table at the bottom about the food tests, so starch is a substance and the reagents used is iodine solution. The colour of reagent is orange at the beginning with a positive test, so it then goes blue-black. With glucose, it's Benedict's solution. It starts off as blue and then it goes to brick red. Okay, so the first mark, okay, is to state between starch and Benedict's. The second is orange and a third is brick red. Attempt this past paper question and 6 marker, pause the video, then we'll go through the answers. Okay, so in this question, Reese was asked by his teacher to set up a light microscope so that he could view some cells at magnification of times 100, and it had 3 objective lenses as 4, 10, times 10, and times 40. In this first part, it says, explain how Reese could view the muscle cells at a magnification of times 100. So it's not enough to say 10 times 10. For these 2 marks, you say use the times 10 objective lens, and then saying the calculation of magnification equals objective lens times the eyepiece lens. So the eyepiece that was given is times 10, and the objective times 10, and that equals 100, okay, for the second mark. So part B, state the function of structure A on the diagram, and that is to bring the object into focus. The focusing knob. Across to this 6 mark question. So the question says compare and explain the shape of the graphs for both types of amylase. So here in the key, we have the solid black line is amylase from humans, whereas the dotted black line here is amylase from bacteria found in hot springs. So in this, we've got our x-axis temperature and then amylase activity on the y. For the 6 marks, I've broken it down. First mark, say more frequent successful collisions with higher temperature due to increased energy for movement. The second, denaturing the enzyme irreversibly changes the active site shape. The third mark, higher temperatures denature enzymes. Fourth, the enzyme activity for both graphs increases. So you can see that on the graph here. They both increase up to their optimum temperatures, and then they decrease, okay, because of that change in the active site. So 5th mark, if you stated the optimum temperature is 37.5, reading off the graph here, Celsius for humans, and then 55 Celsius for bacteria. You get that mark. And then finally, the 6th, enzyme activity stops at 42. You can see that here for humans, and 66 Celsius for bacteria. Okay, pause the video and have a go at these. Okay, so in this question on enzymes, we can see it says the first parts here. Okay, be careful. It says complete the diagram below to show the next stage of the reaction between the enzyme and the two simple molecules from above. So we can see that these fit into that active site, forming this complex. So first mark is that it enters the enzyme, and the second is that they connect. Okay, that they've connected together. The name of this model of enzyme action is called lock and key. And then explain how boiling would affect the action of the enzyme. So it denatures the enzyme, altering the active site shape. So that's for 1 mark. And therefore, the molecules, the substrate, no longer fit together in that enzyme substrate complex. So the second part of this, we have a bar chart showing the relative concentration of different minerals. And we got our key here. So the darker shade of grey, pond water, and then the white is cell sap of alga. Explain how alga obtains the mineral salts from the pond water. What we can see here, the difference between it's always moving from an area of lower concentration to an area of higher concentration. So in this, for 2 marks, the first is to state that it's active transport occurring, and the second is that it's against that concentration energy, or mentioning that it requires energy. Okay, and in part B, so it says suggest given reasons the expected effect of uptake of the mineral salts in the cell sap of raising the temperature from 5 Celsius to 15 Celsius, and it's 3 marks here. So the first mark saying that it increases the uptake as you're increasing the temperature, you're increasing the enzyme activity. And then the third mark is stating that it increases the kinetic energy, so movement, creating more successful collisions, faster rate of reaction. Then in part 2 of B, so adding a poison to the pond water, this is going to result in a decrease in uptake. But again, it's 2 marks, so we must mention that it's going to stop the enzyme action. And that's the end of this video.